and flexing stability. That one. At the end, you talked about system validation and being able to fit your model with sinus rhythm data. When you fit your model with sinus rhythm data, are you able to detect scars to then do ablation guidance? Or do you need to see the heart in AFib to be able to detect that scar region?
It's better in detect scars in sinus rhythm. Actually, we have research work that has not been shown here. It's about processing voltage map on sinus rhythm versus atrial fibrillation. It comes out that processing a voltage map on atrial fibrillation is much more difficult because it's chaotic activation waves, creates a lot of complications in the recordings. So it's actually better to process like a scar map with the sinus rhythm. That's why I said that it's very useful if the physician can cardiovert the patient to sinus rhythm, which by my knowledge, about 50 or more % of patients, they could be cardio converted. Unless they have severe atrial fibrillation, then they will remain atrial fibrillation. Then that would be a more difficult case.
Yeah, so one of your supporting contributions is the ablation guidance.
Yes.
I think that's not supporting. That's one of the major outcomes of your work, which is to guide the surgeon in doing their operation. And did you like validate the guidance with the doctors who are really doing this? Are they agreeing on those readings as well? What is the criteria for selecting those readings?
So this is more towards the future work now.
Okay.
So what we have done is we run our system alongside the clinical system in the operating room. And then we provide like here. So we have our computer squeeze in those many computer screens.
Yeah.
And we will show the physician the additional maps we have created. And we are confident that our map works because the physician liked them a lot.
Okay.
So because when I process it, I will ask physician, is it what you think it is? And then he tells me, yes, it's quite accurate. But we lack of a clinical trial. So so in order to say it publicly, it works, then we have to do more rigorous testing. It probably involves a clinical trial. So that's why I put this in supporting contributions now, because we don't have a solid say about it.
Yeah. You can ask the question.
So we can go through the committee members to ask questions.
Yeah.
Right.
Yes, Dan, please.
Yeah, very nice job. A question about your validation. You showed the difference in those patients that you validated against with atrial tachycardia against sinus rhythm. These atrial tachycardia patients, were they, when you say atrial tachycardia, do you mean that they were in an atrial arrhythmia or just that they were in sinus tachycardia driven by the atrium?
They are during tachycardia. So
but it's a pathologic tachycardia, right? Is that like this is like a focal?
Yeah, it includes mainly focal arrhythmia, and then there is 2 patients with rotor arrhythmias.
Gotcha. Okay, yeah, that was my question, was how it performed on those. And then the next question was, were any of these simulated patients, those redo patients that you suggested, where it can be difficult to sort of redetect where scar tissue has sort of regrown the ability to have electrical conduction?
Well, that's the project I didn't show in the slides here, because that's quite complicated to explain with the confined time. I guess you are asking how easy it is to detect the scar correctly?
Well, no, I guess my question is more, I know that there's sort of minor differences across each of these 8 patients, but whether you saw differences in your
differences in scar
simulation performance based off of the type of patient that was being simulated.
or how many scar the patient has.
Yeah, well, I guess my question is more example for, so was your performance on your simulation when you did this validation worse if a patient had already been ablated once, for example, and had scar?
Oh, actually not. Actually,
Okay.
the eight sinus rhythm here, they are all redo patients.
Okay,
So they have previous scars already.
gotcha. Okay.
And these maps are acquired before the ablation procedure. So these are redo patients, they have previous ablations, and then they come into the operating room. They've been converted to sinus rhythm, or they are in sinus rhythm. And then this is the first map the physician acquires.
I see. Okay, no, that's great. That answers my question. Thank you.
So
Okay.
you know the ground truth.
Right, yes. Hi, Arkady.
Oh, yeah, you want me to ask you a question?
Yes, do you have questions?
Could you explain better how you subtract the QRS in your electrograms? Because you briefly mentioned this about this, but I'm not sure if I fully understood it. So this you somehow calculate, the last part of your,
Yes. So this is an algorithm implemented on not just one electrogram. It implements on 20 electrograms as a set. So this is how the algorithm is built for a set of 20 electrograms together. For example, this is the mapping catheter that contains the 20 electrograms. Why we do that? Because in order to subtract the far field, so the ventricle signal is one kind of far field signal. So when it's far field, then for local regions, they will all pick up similarly. So that's why the first step, we would subtract a medium of these 20 electrograms. That actually have done a lot of work already.
So, okay, Jiyue, may I interrupt you here? So you're calculating medium. You take just one recording, so from one electrode, and try to calculate the median.
Oh, it's the medium of 20. So
of 20. Because
yeah, so for each time frame, each time instance, we have 20 value out of the 20 electrode, and we pick the medium of that 20 value. So we create a medium signal that's for record it for 2.5 seconds. So that means from the 20 2.5 second electrograms, we created a new electrogram called a medium electrogram.
But you are taking in, so you create medium along the kind of, you take one electrogram, and then you go through all electrodes, or you take in one multiple beats, not just a single beat, but multiple.
We just do it on the single beat.
So you do, so instead of looking at one.
Beat. Oh, okay. So I think I
multiple beats along one.
So here, are you saying that there are multiple beats within these 2.5 seconds?
Yes, exactly.
And then we are taking the medium like on the entire 2.5 seconds.
Yes. And then take it out, because you may have very different electrograms and different electrodes, and you will be kind of thrown into this average. You're throwing kind of bad signals. Let's say if you don't have some way of discrimination. Just one question. The other was the one way of kind of avoiding this QRS is differential recordings. So what you're saying, it's not efficient enough using the differential recording. It won't take out the QRS from this.
Yeah, I would say
bipolar, or these are bipolar?
Oh, we are working on unipolar. The algorithm is developed for the unipolar. It's actually Sometimes physicians prefer bipolar, but in some situations that they prefer unipolar. Here, if the physician use bipolar, the QRS may become less in strength, but it might still be there, and that becomes even more difficult to remove because, like, if its magnitude is roughly the same as the endocardium magnitude, then it's even more difficult. That's why we choose to develop an algorithm on the unipolar electrogram, so that the interference is more stand out.
Okay, thank you.
I have another question, Jiyue, but I'm happy to wait if there's questions in the room.
Go ahead. Go ahead.
Can you go back to the slide where you were talking about, you were comparing the results of your fiber-independent model, fiber-dependent, and you had shown a couple of areas, I believe, where your fiber-independent model had missed that.
Yeah.
Okay. So, given what you showed us in terms of your data about how fiber basically only has these local effects, right? Do you see those missed areas from your model as being clinically important to capture?
Oh, I would say that although I'm showing in figure B, it looks like we are missing a large chunk in the distance, but let me explain a little bit more about what this map is. It is the latest local activation locations. So what we are showing is actually the maximum error in the atrium. And in clinical practice, we believe the usage of our heart model is to start from the minimum error regions. And then physicians can do some ablations on that. And then we do another iterations. So we imagine when physicians using this model, we rarely need to access these maximum error regions, because it can be for the next ablation mapping iterations. So while we have more and more ablations done, then the atrium rhythm will become more and more close to sinus rhythm.
I see. So so after every ablation, there's a remapping performed?
Yes, and even for this, let me continue a little bit. Yeah, even for this, like you see a large error of rotor meandering, but the situation here is we simulate a 3 second of rotors, which is rarely happen in clinical. So rotors is a debatable concept in arrhythmia ablation. Nearly half physicians believe in rotors, half physicians do not believe in rotors. For those who believe, the observations of rotors is mainly a couple hundred milliseconds. And at most, they observe up to 3 seconds. That's why we simulate a 3 second. That's also to show the maximum errors we can have with this situation. And I would say that for clinical use, we would, you can see that the beginning of this, they are very near. The importance is to find the original location of the rotor source. So imagine if we have already ablated this region, then probably we don't have any rotor going in this route anymore. And then it's going back to the next iteration of mapping and ablation.
That makes sense. Okay, thank you.
So you're basically saying, if you go back to the other slide, the green localizations, which are the latest occurrence, that correlates to those minima you were showing on those window of interest graphs.
Oh yes, this is a portion of the activation time map, but we restricted the time to the latest 5 milliseconds.
Got it.
So that's where the wave ends. And then you can see that here it is actually because of the geometry. So you do have effects of fibers because it will deviate the activation a little bit, and coupled with the curvature of the atrium. So you can see that patient data with fibers, it can end up in two places because of the curvature of fiber and the geometry. But this is the end of the activation wave. So this is the maximum error we will have.
Is the data source here a simulation? Do you know for a fact where the scar tissues and conduction velocities are for this particular model?
Oh, for this particular one, we don't have scars here.
Okay.
So it's a simulation of multiple different focal arrhythmias originated at different locations. And when we incorporate fiber, we treated that simulation as ground truth.
Okay.
And then we and then we tune our model to that ground truth. And then we compare.
When you look at conduction velocity over this atria, do you find there are conduction velocity errors in this model as well when you fit it? Or is the conduction velocity turning out mostly correct? It's just that the actual local activation time simulations are a little erroneous.
The conduction velocity is highly correlated to the local activation time. So when we have a correct local activation time, we will have correct conduction velocity. Yeah. Because the conduction velocity, you can roughly use distance divided by time. So you have the same distance and the time is the local activation time.
Got it.
Reserve the toughest questions from there. Can you go to slide number 30?
30.
So it maybe was exactly this slide or a few after, but you were speaking about the need to do registration of fibers.
Yeah.
And I think this was ex vivo experiments.
Right.
At the time, what I was trying to understand was sort of the following. If there is no registration, presumably one could get, I mean, what is the purpose of the registration? And how does that affect simulation? And in particular, if you registered, presumably there isn't a lot of fiber direction variation after. And so that's what I was confused as to. You were claiming that you still had an ability to simulate the voltage patterns regardless while capturing different fibers. So I was a little bit confused about this part.
Okay. So first, if you take a closer look at these two different fibers, they are very different. So you mentioned that maybe the fibers are not that different from every patient, but here it is actually very different patient from patient. So the fibers of left atrium, specifically left atrium, has two properties. Two characteristics, I would say. One is that different patients have very different fiber organizations. And then regional, region by region, they are very different too. So in the left atrium, there's not much fiber organization, not like the ventricle. Ventricles, they have a very strong fiber organizations, and they are similar across different patients, but it's not the case for atria. And then for why we register fibers, because we could just use the original atrium and its intrinsic fiber to do that, right? That's another option. But if we go on that route, then we have to somehow develop a way to compare based on different geometry. So how do we calculate the error in between? So after thinking,
perhaps the lack of clarity for me is that you're registering what to what?
I register. So here we have 7 fiber data.
Yeah.
We pick one geometry.
So
Yes, and then it will have its intrinsic fiber, but we still need to register because we are registered on endocardium mesh. So the epicardium still has to deform and register. So this is for one patient, and then for the other patient, you have endocardium, epicardium fibers. You need to deform that into this geometry, and that's what we call register.
But if you choose one of the patients as a template,
Yes.
and that patient, as you just said, there is a lot of variability, and that's not very representative,
Or that
then you might choose patients that has a particular set of anomalies that are not representative of others. So
yeah, that's one of the limitations. And the way we choose that is we reference back to the 7 ex vivo database paper we referenced. And we specifically choose patient 1 because that's what that paper specifically say, that they observe that patient 1's fiber could generalize the best. So that paper, they work on the 7 fiber data, and the goal of that paper, or the conclusion of that paper, is they try to find one fiber atlas that could be implemented on multiple patients, and they open source it so every others could use.
Yeah.
Because, and then we find out this paper, we grab those data, and then we follow them. They say that, okay, patient 1 is most generalizable, then we will use that as our reference too.
actually, that was going to be exactly my next question, but with a slightly different way, which is, I would have imagined that there would be already an atlas that's been built with fiber orientation, and that you would simply be registering your 7 patients to that atlas.
Yes, like there, but as far as I know, there may be several different ex vivo data groups. But the 7 ex vivo one we found that is from the UK group. We believe that's the best we have right now. And like the 2 related works, one of them has implement fibers. They also choose it from atlas.
My second group of questions is, I think it maybe it's like 40 to 44. It's when you begin with the equations, the differential equations.
Begin here.
So in the opening of your presentation, you have 3 different maps. And here you didn't tell us what is U, what is H, how does it relate to the 3 maps, and why once you have estimated the parameters of this model and you can simulate, you can then generate the other 3 maps.
The other 3 maps.
So one was a voltage map, the other
voltage,
was a map, and there was a third one that was maybe
like a coherent map from the current system.
So basically, what are those 3 maps and how do they relate to U and H? And why, if I have U and H, I can generate that?
Okay, so here a little more detailed explanation of the parameter. U is the action potential. So it's the voltage you can measure on the cell.
Yeah.
And then H is the gating variable they have. So that's a transitional of the refractory to active. So they have that H for that. And then we have this constants that's going to change the gating variable H, which controls the shape of the action potential. And this action potential shape is highly correlated to different types of arrhythmias. So it can be narrower or wider depending on the arrhythmia. And
it also depends on which kind of cell it is in which region of the heart.
Yes, we are not that detailed yet, but yes, that's true. And then the simulation will calculate the U values of each time point, which creates like an electrogram thing. I didn't show the equation to calculate the electrogram, because electrogram is like a dipole and next to the tissue, and it's going to be an integration of the entire heart. So integration of about 100,000 U's. That's going to be the electrogram. But for simulation, in order to convert that into the 3 types of maps, we don't really need to calculate the electrogram, because these maps, for example, here is showing the local activation time map. All we need to know is where does the peak of the U values of that point appears, so that value would be the local activation time So each point we have one value, and then color mapping, you get this map. For the voltage map, then we do need to compute the electrogram. When we have the electrogram computed, then for each of the points, we can compute the range of that electrogram, which will be the magnitude. And that value will convert it into a voltage map.
Now, for this model, the parameters of the model, are they pre-specified? Are they learned? And as you just said, they change from patient to patient, region. So what did you do about the parameters?
So what we do here is we test a bunch of simulations, and then we have typical physiological heart behavior, like what the physicians see typically. What's the conduction velocity they see on the left atrium? It's about 0.7 meters per second. So we have, and what do physicians typically see for a tachycardia? It would be cycle length of 180 milliseconds. So those, we tune the taus to match a typical left atrium. That is, like for sinus rhythm, is typical for sinus rhythm, tachycardia typical to that. And then we are not going to tune it anymore.
Yeah, that was going to be exactly my next question.
Right.
So there was a section of your work on tuning, and you were tuning the D.
Yes, the D, the diffusion.
And I imagine exactly as you just said, that you would begin with a set of predefined parameters for the model, as well as expected values for D.
Yes.
And then in the same way you did the tuning, you would optimize over both D as well as the parameters of the model to tune into that patient. Why did you tune only the D but not the other?
So the most important thing here is we have restricted our model to work on up to tachycardia. So there are different complexities of arrhythmias. We would say, started from a healthy heart, we have sinus rhythm. Then we have tachycardia. Then we have the real difficult atrial fibrillation. To simulate accurate atrial fibrillation, we would inevitably tune the tau values, because that's very important. And we haven't got to that part yet. So what we are is before that, all the rhythms before that, the tachycardias, like the re-entry circuits. And for those, we can implement a typical set of tau values, because those cycle lengths are not that short. So that the most important factor would be the conduction coefficient, because the action potentials, they do not get too tight to start to influence each other.
And for the tuning of the D, what is the initial values? Is it obtained from that preoperative data that you have from certain patients, or is it preset values?
It's also coming from some experiments where we find a D value that's going to simulate a typical healthy heart. So that would be our initial guess. So we put in a healthy heart diffusion coefficient as the initial guess.
And so this was a long time ago, but people did think, maybe this was a research project and not so clinical, but people did have preoperative diffusion sensor imaging, from which they would extract fibers and use that to initialize the model. So this is not used today in the clinic still?
No, it's not clinic. The data set, atlas we have are from the DTMRI.
Yes.
But still, for high resolution, like for enough resolution, they take tens of hours still up to today. But the paper is about 2020.
Well, I would argue that there's probably a trade-off. There are ways of scanning that is
faster,
that you can do in less than 1 hour. But then it's just less accurate. And the question at the end of the day, is that more accurate than the D and 2 that you do?
Yeah, we would like to work on a heart model that's with the fibers, because that's less less compromised.
No, it's highly accurate. It has to be excluded. So it is in vivo. And my last question is, there was a point in time where you said you were looking for the minimum of some map in order to determine the location of where to ablate.
I think that's...
I just didn't understand what is the map that you use and how does it relates to your model? Is it the minimum of the voltage map, or is it the minimum of an activation map that is derived from the voltage?
I think you talk about
Yeah.
the slide here. This is activation.
In other words, how does one compute the ablation guidance map from whatever the voltage that comes out of it?
Also, to answer that, I will go here first. We have the ablation guidance map. It's not one map. It's a variety of different kinds of maps for different usages. One map is one feature. So here is a listing. We have this many features. They can be converted into a map. And for the slide here, I'm showing is the activation time map. And this map uses color to mark the sequence of activation. The earliest is marked red. The latest is marked blue. So there is a type of arrhythmia called focal arrhythmia. And to find that location is to find the earliest activation location. So that's why we are going to try to find a spot of red. But in clinical practice, like the technician could restrict their time of interest in the screen. It's adjustable. But then, like there are about 2,000 electrogram recordings. No matter how they adjust, if the activation, if the rhythm has some overlap or have some like too fast or too slow for some situations, they will never have a clean cut. So it's very easy to get into some map like this. When you're trying to look for the red, then it doesn't make sense because I trying to find a focal red, but it's a large areas of red, and then they have to spend time to readjust it again. And what I'm saying here is, why not just implement a computer to do that? It's not that difficult to do. Find local minima.
But that was my question. How is that surface generated?
This, so for this particular example, let me explain this first and then go to the atrium. I choose a slab here because then I'm able to plot this. So you know that these values on the z-axis are the activation time values. So you have x, y, and the value. Then you find the local minima. On the atrium though, then it becomes 4-dimensional now. So for every mesh location, you will have that associated activation value. It's going to be also just need to find like a local minima. Then it would be the focal space.
Yeah, so. Okay. Any more questions? Anyone or from the committee online? Okay, ask them again. After I will go.
Hi, Arkady and Dan. Yeah, additional questions?
No.
So yes, I will ask you for an applause. Okay, switch off the recording.
Okay.
Leave the people.